this vlog, I will discuss about protein synthesis, which is transcription and translation. This amazing artwork shows a process that takes place in the cells of all living things. The production of proteins. This process is called protein synthesis, and it actually consists of two processes: transcription and translation. In eukaryotic cells, transcription takes place in the nucleus. During transcription, DNA is used as a template to make a molecule of messenger RNA. The molecule of mRNA then leaves the nucleus and goes to our ribosomes in the cytoplasm, where translation occurs. During translation, the genetic code in mRNA is read and used to make a protein. These two processes are summed up by the central dogma of molecular biology. Transcription. Transcription is the process by which DNA is copied to mRNA, which carries the information needed for protein synthesis. Transcription takes place in the two group steps. First, pre-messenger RNA is formed. With the involvement of RNA polymerase enzymes, the process relies on Watson-Crick-based pairing, and the resultant single strand of RNA is reverse complement of the original DNA sequence. The pre-messenger RNA is then edited to produce the desired mRNA molecule in a process called RNA splicing. Formation of pre-messenger RNA. The mechanism of transcription has parallels in that of DNA replication. As with DNA replication, partial unwinding of the double helix must occur before transcription can take place, and it is the RNA polymerase enzymes that catalyze this process. Unlike DNA replication, in which both strands are copied, only one strand is transcribed. The strand that contains the gene is called the sense strand, while the complementary strand is the antisense strand. The mRNA produced in transcription is a copy of the sense strand, but it is the antisense strand that is transcribed. Ribonucleoside triphosphate align along the antisense DNA strand. With Watson-Crick-based pairing, RNA polymerase joins the ribonucleotides together to form a pre-messenger RNA molecule that is complementary to a region of the antisense DNA strand. Transcription ends when the RNA polymerase enzyme reaches a triplet of bases that is read as a stop signal. The DNA molecule rewinds to reform a double helix. RNA splicing. The pre-messenger RNA thus formed contains introns, which are not required for protein synthesis. The pre-messenger RNA is chopped up to remove the introns and create messenger RNA. Alternative splicing. In alternative splicing, individual exons are either spliced or included, giving rise to several different possible mRNA products. Each mRNA product stood for a different protein isoform. These protein isoforms differ in their peptide sequence and therefore their biological activity. It is estimated that up to 60% of human gene products undergo alternative splicing. Alternative splicing contributes to protein diversity. A single gene transcription can have thousands of different splicing patterns and will therefore code for thousands of different proteins. A diverse of proteome is generated from a relatively limited genome. Reverse transcription. In reverse transcription, RNA is reverse transcribed into DNA. This process catalyzes reverse transcriptase enzymes allow retroviruses, including the human immunodeficiency virus or HIV, to use RNA as their genetic material. Reverse transcriptase enzymes have also found applications in biotechnology. Steps of transcription. Transcription takes place in 3 steps: initiation, elongation, and termination. Initiation. Initiation is the beginning of transcription. It occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. This signals the DNA to unwind so the enzyme can read the bases in one of the DNA strands. The enzyme is ready to make a strand of mRNA with a complementary sequence of bases. Elongation. Elongation is the addition of nucleotides to the mRNA strand. Termination. Termination is the ending of transcription. The mRNA strand is complete and it detaches from DNA. Processing mRNA. In eukaryotes, the new mRNA is not yet ready. At this stage, it is called pre-mRNA, and it must go through more processing before it leaves the nucleus as mature mRNA. Processing may include splicing, editing, and polyadenylation. These processes modify the mRNA in various ways. Such modifications allow a single gene to be used to make more than one protein. Splicing. Splicing removes introns for mRNA. Introns are regions that do not code for the protein. The remaining mRNA consists only of regions called exons that do code for the protein. Editing. Editing changes some of the nucleotides in mRNA. For example, a human protein called EPOP, which helps transport lipids in the blood, has 2 different forms because of editing. One form is smaller than the other because editing adds an earlier stop signal in mRNA. Polyadenylation. Polyadenylation adds a tail to the mRNA. The tail consists of a string of As. It signals the end of mRNA. It is also involved in exporting mRNA from the nucleus, and it protects mRNA from enzymes that might break it down. Translation. Translation is the second part of the central dogma of molecular biology. It is the process in which the genetic code in mRNA is read to make a protein. After mRNA leaves the nucleus, it moves to a ribosome, which consists of rRNA and proteins. The ribosomes read the sequence of codons in mRNA, and molecules of tRNA bring amino acids to the ribosome in the correct sequence.